who is Dr. Ahmed Badran. Did I say that right?
Yeah.
Badran, cool. Who is associate professor in the Department of Chemistry and Integrative Structural and Computational Biology at Scripps Research Institute. Ahmed earned his bachelor's in biochemistry and molecular biophysics from University of Arizona in 2010. He completed his PhD in chemical biology at Harvard University in David Liu's lab, where he helped lay foundations for modern continuous directed evolution and genome engineering technologies. 2017, he joined the Broad Institute as a Broad Fellow and postdoc, where he continued developing these methods to rapidly evolve biological function in living systems. Ahmed started his independent lab at Scripps in 2021, where he has built a research program that sits at the intersection of chemical biology, synthetic biology, directed evolution, genome editing, and of course, genetic code expansion. Unifying theme of his work is he tackles systems that are traditionally considered too essential or too central to engineer, like translation or carbon fixation, and developing new ways to interrogate them without compromising cellular viability. His research area spans a few different areas that are interconnected, including re-examining biological carbon capture by engineering ribosome and carbon fixing pathways. They're also developing next generation antibiotics using engineered translation machinery and automated evolution. And for this audience in particular, of course, his lab is also pushing the frontier of genetic code expansion as a core technology by developing new GCE platforms that operate without host genome modification and integrating GCE with in vivo evolution to discover artificial enzymes and genetically encoded molecular glues. Ahmed has authored over 30 high-profile publications in his short career. He's won numerous awards, including the NIH Director's Early Independence Award and the Presidential Early Career Award for Scientists and Engineers, which, when I looked that up, was described as the highest honor bestowed by the U.S. government on outstanding scientists and engineers beginning their independent career. So that's nothing short of very, very impressive. So we're super excited to have you here with us today. Ahmed, please take it away.
Thanks, Rick, for that really kind introduction. As Rick highlighted, my lab works in a number of different areas. So I'll share with you a little bit of our work on the genetic code expansion space, and then I'd be happy to answer any questions on anything else at the end. So my lab for a few years now has been interested in sort of methods development to create unnatural macrocyclic peptides. This is a bit of a deviation from the title that I originally submitted, but I think segues really well from June's fantastic talk. So just to orient us all, of course, we know of macrocycles in many different contexts. Most of us have thought about these in the context of natural products, particularly ones that are produced by by non-ribosomal peptide synthesis. But of course, there's a pretty large variety of different strategies to make macrocycles, including post-translational modification of RIPs. And in the more sort of synthetic biology spaces, cyclops and phage display have been used to develop and evolve and functionally mature cyclic peptides for specific bioactivities. So where we really got interested, and I'm going to skip a lot of the sort of foundational stuff in the genetic code expansion space because June did a really great job. Our interests really derive from the fact that a lot of these bioactive natural products or cyclic peptides are built predominantly using the 20 canonical amino acids. And RPSs are, of course, an outlier to this, but they have historically remained recalcitrant to engineering to really change the scope of biomolecules that they make. So as June highlighted, a lot of our work leverages maybe 3-decade-old technology now in genetic code expansion that was established by Pete Schultz and Dieter Sohl and many, many others that shows that you can engineer these tRNA synthetase pairs and establish new amino acid scope, or more increasingly in the modern era, non-canonical monomer scope that can be incorporated through translation into polypeptides. And the scheme that's shown here is really that you can use directed evolution to engineer these active sites and change their amino acid scope. And this has been applied extensively, including by many on the call, especially at the GC4All Center, to really establish tRNA synthetase pairs that incorporate nature building blocks. So fundamentally, the sort of issue that we started to tackle is not necessarily on the chemistry side, as June talked about the way that you incorporate the monomers, but rather on the codon problem. And what I mean by this is that if you think about the genetic code as being like a pre-assigned operating system, fundamentally what genetic code expansion does is it goes into that operating system and it changes the correspondence between a codon and a building block, an amino acid. And so one of the earliest ways to sort of usurp information from that genetic code was to take advantage of stop codon suppression, of AMBER suppression, that takes advantage of the fact that UAG in E. coli and humans and some other organisms is the lowest usage stop codon. So you can sort of convince the ribosome to translate proteins by having a tRNA that reads that triplet codon. But increasingly, there are other strategies that have been developed, including whole genome rewriting, development of synthetic nucleobases, and a little bit more recently, developments in quadruplet codons, which are a little bit of a weird space because what is really fundamentally happening with these tRNAs that have an expanded anticodon loop is that the ribosome is, in a certain instance, reading a 4-base code rather than the natural triplet 3-base code. So at the outset of our efforts, we tried to understand what a lot of these processes brought to the table and the ways in which they could be optimized. And fundamentally, the things that we concluded is that there's a lot of attributes that we might want, especially for multiplex genetic code expansion, but that many of these had too high of an opportunity cost to really optimize. But quadruplet decoding through these 4-base codons could potentially be a really phenomenal approach to incorporate multiple non-canonical amino acids if we could somehow understand why this process was so particularly inefficient. So just to orient you all, I'll give you an extremely high-level summary of how many people in the field think about quadruplet decoding. This is a severe oversimplification, and not everyone agrees with this, but I think it'll be helpful to sort of think about it in this context. So imagine the ribosome is translating an mRNA where you see those 4 blue pins, that's a quadruplet codon. The A, P, and E sites are the sites of the ribosome. So at the first step, the ribosome sort of meets with this 4-base codon in the A site. And the first bad thing that can happen is they can put a sort of natural triplet tRNA at that spot. This frame shifts everything downstream and doesn't make the product that you want. So this is something we don't want. If you provide a quadruplet decoding tRNA that's been amino acylated by its cognate synthetase, the ribosome can sort of put this in the A site and try and mix and match those 4 bases to make sure that it's a good fit. But a lot of the time it spits that out because it doesn't really fit very well in the ribosome. With some frequency, it proceeds to the right. It moves that tRNA into the P site. It forms a new bond between the growing polypeptide chain and the incoming amino acid. And then once it does this, it can sort of sample the space to maximize that quadruplet interaction. But if it doesn't and sort of retains that 3-base interaction exclusively, a triplet tRNA can come in the A site and sort of take advantage of that last base of the quadruplet codon that now appears as a triplet. Again, this results in a frame shift of the downstream protein, which isn't particularly meaningful. But going back to the top right, if it does that, then the next triplet tRNA comes in and the frame of the protein translation is restored. And so this is sort of the correct product that you want to be able to make. And what we wanted to do is to increase the efficiency of these blue arrows to drive it all the way to the bottom right. And so at the outset, there are a number of limitations that we and others have recognized. The decoding by quadruplet tRNAs is quite inefficient. There's competition with host tRNAs. There may or may not be local sequence requirements that dictate the efficiency of this process. And of course, the fact that you have to sort of push it back into the triplet frame immediately after the quadruplet decoding event. So at the beginning, we decided to look first at tRNAs and the contribution that they would have in an otherwise natural translation setting. So it's a natural ribosome. The tRNAs are all available in the cell, and we just provide a quadruplet decoding tRNA. But also we create these reporters where there's a quadruplet codon in the middle of the gene of interest. These are just too easy to monitor reporters. But what we found is that there's a number of interesting relationships, most of which are bad for the applications that we want to have. The first is that if we take a number of different scaffolds, so these are tRNAs that encode the amino acids that you see at the top, and engineered their anticodons to read the codons that you see at the bottom on the x-axis of this graph, most of the time they don't work. And the times that they do work, you can see on the y-axis, we're relating this to natural triplet translation, which is 100%. And so in general, the efficiencies are incredibly poor for a single decoding event in an otherwise triplet gene. You can sort of flip this paradigm and use something that looks like an amber codon, which is this UAGA quadruplet codon, for different tRNA scaffolds, and find that, again, most of the time they don't work, and when they do, it's quite inefficient. So this is where we got initially started. We know that there's a lot of these tRNAs that exist in the literature. In general, if you try to engineer them, the hit rate is incredibly low, and it's not really clear what makes a tRNA quite functional in this respect. So the first question we asked is, can we evolve these quadruplet decoding tRNAs, or qtRNAs, to be higher efficiency? And for this to work, the tRNA has to play a pretty active role in that frame maintenance during translation to invoke that frame shifting. So to do this, and I'll sort of skip exactly how this works, but we piggybacked on a technology that I helped develop as a graduate student called PACE, or phage-assisted continuous evolution. PACE automates directed evolution to reduce the round of directed evolution from many days down to about 10 minutes. And this allows us to evolve tRNAs very, very rapidly. So these evolutions we do only in about 24 hours, and we allow the tRNA to evolve better decoding for its cognate quadruplet codon. And what I hope you can see is that for the different scaffolds, again, that are sort of indicated at the top, the ones we engineered to have a quadruplet anticodon, in this case, it reads the amber codon UAGA, they're quite inefficient, but only after 24 hours of PACE, we can evolve variants that are shown in red that have much higher decoding efficiencies. We didn't know exactly where these mutations would show up. When we sequence things, the mutations are all over the place. Sometimes you can rationalize how these might improve quadruplet decoding. For example, in the serine decoder, many of the mutations show up near the anticodon loop, and maybe you can invoke, a better fit in the ribosome or a slightly altered structure. But in other cases, they're very distant from the decoding that's happening, and so it's actually not quite clear how these tRNAs are able to do this. But fundamentally, the tRNAs are able to read their quadruplet codons more effectively. And they show many hallmarks that would suggest that we can use this in a multiplex setting. So for example, if we make many tRNAs that put in the amino acids that you see on the left, they have anticodons corresponding to the codons that you see here. And then we ask, can they read the quadruplet codons that we see on the bottom of this figure? And what we find is that generally the diagonal lights up, which is the sort of cognate codon-anticodon interaction. So they are mutually orthogonal. All the ones that you see at the bottom here, they just read UAGA, and so they only light up on the right-hand side. They also show a really interesting property, which is processivity. So if we make a reporter that has multiple quadruplet codons immediately back to back, our PACE-evolved qtRNAs can actually show relatively efficient processivity in decoding these quadruplet codons. Again, suggesting that the tRNA plays a really active role in maintaining the correct, from its point of view, the correct frame during translation, which in this case is multiple quadruplets back to back. Okay, so we got pretty excited at this about this and wondered, can we start to do multiplexing? And so what I mean by that is a typical reporter that is encoded by triplet codons, and then at specific positions we put quadruplet codons that are mutually orthogonal and ask, can we make a protein that has multiple different quadruplet codons in it? So these are the efficiencies of single incorporation events. The amino acids are shown in the bars. The codons that they're reading are shown at the bottom. And the efficiencies are pretty good for a single quadruplet codon, but as you start to sort of mix and match these, you get very, very close to the limit of detection of the assay, which in this case is making GFP. And this suggests that there's something else that's going on. And then maybe it's not so surprising that the efficiencies start to drop quite precipitously, because from the perspective of the ribosome, it doesn't know what code you're trying to invoke. And so at every step, it's sort of reading a 3-base code, and then where you define it, it's supposed to read a 4-base code, but as far as it's concerned, that's still a 3-base codon, and so it can proceed in that sort of incorrect frame a significant fraction of the time. So at this point, we took a step back and tried to survey the literature to understand what parameters could influence this process. And to cut a long story short, at this point in time, every single attribute of the translation apparatus in live cells had been explored with respect to its contribution to quadruplet decoding, meaning we and others had evolved tRNAs, the ribosome had been engineered, elongation factors, synthetases, but somehow the mRNA had sort of eluded the study of its contribution to quadruplet decoding. And this raises a particular concern, which is if you have a gene of interest, you want to put a quadruplet codon in it, you don't really have a lot of possibilities of changing the mRNA because you fundamentally want the protein to be a specific sequence. But where you do have the ability to change things is in codon usage. So by taking advantage of the redundancy of the genetic code, we asked a very simple question. How does codon usage near a quadruplet decoding event influence decoding efficiency? And so to do this, we did a very simple experiment. We created a library in the middle of the gene for GFP, where the 5 amino acids before and after a quadruplet decoding event were altered such that every codon corresponding to that amino acid was represented in this library. And then we made all possible combinations of this, and we had a sort of negative control where we put the amber triplet codon at this position. We put all of that in the GCE strain because we're making GFP, we can sort everything by facts. And we took the highest performing subpopulation and the lowest performing subpopulation and just asked, compared to the unsorted library, what is the distribution of codons that occurs in the high GFP versus low GFP? So I'll just show you the most important thing that we found, which is at what we call the +1 position, which is immediately downstream of the quadruplet decoding event. In GFP, at least in superfolder GFP, this happens to be an isoleucine. Isoleucine has 3 codons, AUA, AUC, and AUU. So the library has a roughly 33% distribution going in. If we sort any of these bins, the distribution is unperturbed, which means that the amber codon doesn't really care about the identity of that position. But if we look at quadruplet codons, especially for ones that, noting that we've changed the codon here is UAGA, here's AGGA and AGGA, and that the amino acids in all cases are different, the relationship that we see is very similar. Specifically that the blue things are de-enriched in the bottom bins and enriched in the higher bins. And it turns out that the blue codons are actually the high usage codons and the gray one is a low usage codon, meaning its tRNA is relatively non-abundant in the cell for the low usage tRNA. And we reconcile this in the context of the thing that the sort of schematic that I showed you earlier, where we hypothesize that the ribosome is sort of going back and forth between trying to read that quadruplet decoding event. And if the next amino acid tRNA comes in very quickly, then it forces the ribosome to commit to that quadruplet decoding event. We had some other findings in particular that sort of relate to crosstalk between codon and anticodons, but I'll sort of just skip that. I'll invite you to check out the paper if you want to learn more about it. Okay, so at the end of this, we concluded that we had to do something relatively aggressive. So to minimize crosstalk between triplet codons and quadruplet codons, we don't want sequences that look from one camp to look like the other camp. And so what that means is we have to remove triplet codons that look like the quadruplet codons that we want to use. Then the other thing is we've already identified that high usage codons immediately downstream of the quadruplet codon increases translation efficiency. So to ensure that we can put amino acids wherever we want to put them, we hypothesize that we can create this compressed codon framework where every amino acid only gets one codon, and that codon is a high usage codon. And that eliminates sort of the competition between these two codes and ensures that we can put in a quadruplet codon wherever we want. So this is just the codons that we chose corresponding to each of the 20 amino acids. The left figure is just showing you that the system works quite well. And then when we recode it on the right, it doesn't seem to take any sort of hits. And it is dependent on the non-canonical amino acid that's being added. Okay, so now it looks like we have a foundation to really increase the efficiency of quadruplet decoding. How far can we take this? So building on a lot of work from the literature, we nominated 5 mutually orthogonal tRNA synthetase pairs. Each one of these boxes is a different set. They don't crosstalk. 3 of them are based on pyrrolysine synthases. One is a tyrosine synthase and one is a tryptophan synthase. Each one has its own tRNA. And in some cases, we sort of had an intuition about what codons would work well, and in others, we didn't really know. So we tried to evolve these in multiple contexts. And fortunately, what we're able to find is many of these systems were quite readily evolvable. I'll just walk you through how to interpret these figures. So everything is color-coded, similar to what I showed on the previous slide. The first set of 2 bars is plus or minus non-canonical amino acid for the starting system. The second set of two bars is plus or minus non-canonical amino acid for the evolved system. And then the structure of the tRNA is shown with the mutations that we evolved shown in the respective colors. And what I hope you can see is that they all work pretty well, and importantly, all show a really large dynamic range, plus or minus non-canonical amino acid. But the most important thing is the y-axis. So everything is still normalized to triplet decoding, and 100% is just the normal wild-type GFP that you make if you're just making GFP. And so hopefully what you can see is that the efficiencies of these systems are incredibly high, that we can access relatively efficient incorporation of these non-canonical amino acids using quadruplet decoding rather than the traditional AMBER approach. Another benefit of this is that these synthetases have relatively promiscuous active sites that accommodate non-canonical amino acids that have structural similarity to one another. And so we nominated the 40 or so amino acids that you see here, whose side chains are on the right, and screened them through all of the synthetases that we've developed. And what we found is that many of these synthetases have sort of focused amino acid scopes. And what this means is that with the same DNA sequence, you could add different non-canonical amino acids and create different products on the back end using the same tRNA synthetase pairs. Okay, so I told you at the beginning that the motivation was macrocycles and that genetic code expansion could really enable this. And now we have systems that all read different quadruplet codons, as you can see here on the y-axis. They all show really good efficiency and they don't crosstalk. So can we actually start to make macrocycles? And here's where we took advantage of a complementary system called Cyclops. I don't remember exactly what the acronym stands for, but basically the way that this works is it takes advantage of a really weird class of protein called inteins. These are the protein equivalent of introns. They self-excise from proteins, and in a quirk of how they work, if you flip their orientation of these two proteins, the intine will actually cyclize the peptide that's in between. And so what that means for us is that we can engineer a sequence like what's shown in the box here. We can assign each of these to a quadruplet codon and add different non-canonical amino acids, and the intine will splice that for us to create the macrocycles that are shown at the bottom. This is just to show you broadly how the system works. We encode the different tRNAs, we encode the intine, we provide the synthetase and the non-canonical amino acid and just ask, can the macrocycle be made? These are just positive controls with canonical amino acids showing you that we can see it really robustly by SCMS. If we mutate the enzyme, then we see nothing. The yields are really excellent as well, as I'll tell you in a couple of slides. So we put all of these things together. These are our improved synthetases and our evolved tRNAs, and just ask, can we start to make these things that look increasingly like natural products? So here's some of that data. So just to walk you through on how to interpret this, each macrocycle structure is shown. The highlight in color is the non-canonical amino acid. The color code corresponds to the synthetase that incorporated it. And the boxes show with the non-canonical amino acid on top, without the non-canonical amino acid in the middle, and when this intine is mutated to be inactive on the bottom. And what I hope you can see is that the top box always shows a really nice peak that is lost in the bottom two boxes. The systems are actually quite processive again, so we can incorporate multiple of the same non-canonical in the same macrocycle, and we can even start to multiplex to make these molecules that look increasingly natural product-like. So the final thing I just wanted to show is that the yields are also quite high and robust. So we synthesized all of the macrocycles that you see here by solid phase peptide synthesis, which in the best case scenario, this is from probably the standard peptide synthesis company, which is GenScript. The shortest time frame was 6 weeks, and the longest time frame was 11 weeks, and we could make these in a day. So I think it just shows you the power of genetic code expansion. So at the top is that solid phase peptide synthesis standard. The bottom is what we get from the decoding, and by creating a standard curve, we can actually quantify the concentrations where we get 1 to 10 micrograms only from half a mil of E. coli culture. So there, we can easily sort of increase this to be well in this concentration regime of natural products. So with that, I'll just sort of summarize the way we're thinking about this problem now. And we've done a lot of work in this space to show that we can evolve tRNAs to improve the way that they read the quadruplet codon. We can make these judicious design choices on how we select codons. The mRNA codon compression improves these local sequence requirements, and also it allows us to get around the low use competition with low usage tRNAs when we recode these mRNAs. So hopefully with all of that, I've convinced you that some really simple experiments can teach us a lot about how the genetic code works and how we can sort of take advantage of that to incorporate new to nature building blocks, and that the tRNA actually does play a pretty dominant role in how that genetic code is read, including probably enforcing a significant degree of control on the step size of translation. And then we can combine these resources with others like Cyclops to create really exciting looking natural products that we're natural product like molecules that we are actively exploring applications for. And I should say the image that I'm showing you on the top right is from unpublished data that we have where we can now multiplex this and incorporate 5 unique non-canonical amino acids in the same polypeptide chain, even when they're back to back, like what's shown in this sequence. So we're applying this in a number of different directions. My lab is really good at developing biosensors for a number of enzyme activities, and we're actually exploring how we can use these macrocycles to discover activators or inhibitors of enzyme activity in cells, but also layering this with other backbone modified monomers that we can incorporate or potentially sidechain type intermolecular chemistries to create bicycles or tricycles. So with that, I'll just thank the people that did the work. All of that published work was done by Alan in the lab, who's now leading his own group in Ireland. Cindy has now taken over this project and really pushed it to much higher extremes where, like I said, we can incorporate 5 unique monomers in the same polypeptide. The drone video that's shown on the bottom left, actually, I didn't pay for, but was part of a 100-year anniversary at Scripps that the Institute paid for, so you can see what my lab looks like. Everyone's diligently at work working, wearing lab coats for the first time since I've been at Scripps. I'd like to thank my funding sources for supporting our work and you for your attention. I'd be happy to take any questions.
That was great.
Ahmed, I've got a quick question for you.
Ryan, would you like to ask a question?
I would
Yeah,
like to ask a question. Sorry, I've got to run really quick. Really exciting advances. I love the pushing the boundaries here to the next level with the quadruplets and the cyclization. It's obvious from the natural code that tRNA post-transcriptional modifications are a heavy player in many aspects of orthogonality and efficiency. In the quadruplet space, it would seem unbelievably obvious that this would be a major consideration, considering the post-transcriptional modifications on that fourth base that happens in so many systems. How much are you evaluating the post-transcriptional modifications on your tRNAs and evaluating how to manipulate that?
Yeah, so I've actually tried to initiate no less than 5 different collaborations to answer this question. As I'm told, there is a heavy experimental burden to assess post-transcriptional modifications that you are not confident are there from some other assays. So sort of the de novo identification is really tricky. So I would love to know that answer, but we have not really invested a lot of effort in it. But what I can tell you is that in related systems where we take tRNAs that incorporate canonical amino acids and evolve those to be quadruplet decoders, we can get them to be quite efficient in cells. If we then go and use them in pure systems, they don't work. But if we use them in lysates, they do work. And so that right off the bat, I think implies that post-transcriptional modifications are really important. Probably just generally to tRNAs, but definitely our quadruplet decoding tRNAs.
Right.
But I don't know exactly how that's happening.
Let's connect offline and we can talk about some ideas.
Yeah, I'd love that.
All right, great.
That's right.
I have a question about, so you're really focused on the generation of your cyclic peptides and, making full-length GFP. But have you looked, obviously you're doing these in ncAAs and in cells, like how much now with a quadruplet are you incorporating your non-canonical and just the native proteins in the system that you're working with?
Yeah, that's also a great question. I think we have not looked at this, but I can tell you some superficial pieces of information that might be relevant. So we don't see toxicity from the amino acid in a single setting or when we sort of multiplex them. So we can add, 5, 6 different non-canonical amino acids at 1 to 10 millimolar concentrations and the cell seems like it's fine. That probably is a commentary on the synthetases in the cell using the amino acids we've provided. But there's probably also, you would expect, toxicity coming from if the tRNAs we make are all amino acylated, they all read quadruplet codons, those should read something in the genome, and that should result in toxicity. So one of the things that we've been really surprised about is we don't see evidence of that. We see maybe a minor reduction in cell density at the end of the experiment, but we attribute that more to the fact that, especially when we're multiplexing these high numbers, that we have to make the different synthetases and the tRNAs and the fluorescent proteins. So we think it's sort of a burden issue. We have shown that we can, and this is also an unpublished work, we can evolve these synthetases to make them better catalysts somehow. We don't exactly know what's improving. It could be solubility, stability, catalysis, who really knows? But that allows us to express less of them. And when we do that, then the sort of burden that we see goes down, but the efficiency of incorporation into our protein increases. So it's a little bit surprising, actually, that the cell is able to deal with this so well. But my hypothesis is that because the quadruplet codons are based fundamentally on low usage triplet codons that are just extended by a single base, it's possible that those codons don't exist in essential or highly expressed genes. So we don't see as big of an impact at mistranslation of those host proteins compared to our mRNA, which is being highly overexpressed, and that sort of sponges up those tRNAs for the preferential translation of that protein. At least that's how
theoretically, right? It would cause a frame shift,
right,
in any sort of, but maybe the E. coli can just deal with this.
Yeah, that's sort of, but I think, there's probably really nice experiments that can be done here where at least if the amino acid has some handle that we could pull on, like maybe a click handle, we could sort of pull everything out and see what's actually getting on.
Yeah, and then you would just see basically the truncated protein.
Exactly.
But yeah, that's really cool. I mean, in the end, if it doesn't cause a reduction in cell health, I mean, for your purposes, doesn't matter.
Exactly.
Yeah, but more of a kind of an interesting thing to look at.
Yeah.
Doesn't look like there's any questions online. Any last minute questions?
I can do a real quick one. Probably one. Oh, Andy. I'll do one real quick. I think it's probably a question you get all the time, Ahmed, which is, all those rules about codon context for these quadruplet decoding events. That's all in E. coli. Do you think they transfer also to eukaryotic systems as well, or is there any? I'm sure you get that all the time, but I'm just curious.
Yeah, I do get asked this question all the time. My answer is I need someone to come and try that out. We haven't actually done it because 90% of our work is in E. coli.
Yeah.
But yeah, it's hard to tell. I wouldn't be surprised if there are related rules, but also translation in humans and eukaryotic cells is just so much more complicated.
Yeah.
We are also making synthetases that are more efficient. I think those would probably plug in much easier because you wouldn't need as much.
Right.
And maybe our tRNAs don't have to be expressed as much because that's also another barrier where you have to have, maybe like 50, 60 copies in human cells to get it to go well.
Yeah.
So I want to say my short answer is maybe, but we haven't tested.
Cool.
We could probably help you with that, Ahmed, if you're interested in looking at that.
I think, yes, that would be phenomenal. I would love to explore that.
Andy?
Yeah, just quick. I imagine happens, but the +1 site, high abundance tRNAs work well, and low abundance tRNA codons don't work well, right?
Right.
And if you just overexpress the low abundance tRNA, do you show that it works now well, just to prove that really is the issue?
We've done that in one case, and it correlates positively, but the impact is not as large. We haven't extensively studied it, but I think the relationship is correct. We've done sort of the opposite, where we take just normal E. coli codon optimized GFP, which is mostly high usage codons, but it has positions that are low usage. We try to incorporate a quadruplet codon there. We get some activity. Then we change the +1 position to high usage codons, and that activity goes up. So it doesn't really seem like it's context dependent as long as you enforce that rule.
Okay.
Yeah.
Thanks.
Yeah.
Fascinating stuff.
Thank you.
Cool. One last question. Have you looked at just ribosomal pausing during some of these? So the speed of translation that you're getting and incorporation. So it's not just the physical interactions at the A site, but kind of the +1 position is shown to change your ability to suppress stop codons, for instance, right?
Right.
And so is it just that the ribosome was pausing and allowing this to have a high efficiency, or have you looked at this?
I actually think it's the opposite. I think the faster the ribosome goes, the higher the efficiency is. And we only have one piece of evidence that suggests this, which is ribosomes generally sort of ramp up the speed of translation from the start codon to somewhere, and then they're sort of constant, and then they ramp down afterwards. If you look at the incorporation efficiency, depending on where it is in the mRNA, it looks exactly like that. It sort of comes up, it's like poor at the very beginning, then it comes up, then it plateaus, then it comes back down. So that suggests that it's correlated with translation speed, which to me suggests that the process of the quadruplet decoding is a step that you don't want to give the ribosome too much time to think about, I think. But we haven't done the biophysics to really establish why that's the case.
Yep. Cool. All right. I think that that's it. I'd like to thank the speakers again for giving some great talks.
Thank you.
And we look forward to seeing you all in a month.